Hi everybody, welcome to the Topics in Neurobiology course. My name's Kate Jeffrey and I'm a behavioural neuroscientist and I'm interested in the biology of memory, and that's what this course is about. So the idea is to to look at the historical evolution of ideas about memory and how neurobiology has started to unravel the mechanisms of memory, how it all happens in the brain. So it turns out that memory and representation of space for navigation are very, very intimately connected. So for some reason, the evolution of the memory system and the evolution of the navigation system have really gone hand in hand. And that makes a certain amount of sense because the most important things that happen to you happen in a place, and it's usually meaningful to know where things happen so that that can influence your future behaviour. So we're going to look at that in this course. I'm going to start with the history of ideas about memory and ideas about space, and then we're going to work our way through and then get to the neurobiology. So the scientific study of memory really began almost 150 years ago with the early development of psychology as a discipline. And one of the people who has been incredibly influential in developing these ideas is William James, who's sometimes called the father of psychology. And he had some really insightful ideas. And one of the things that he sort of intuited about memory is that it's to do with the connections that are forming in the brain between representations. So he thought of these connections as paths in the brain. So he wrote, "Memory being altogether conditioned on the ability to excite brain paths, its excellence in a given individual will depend partly on the number and partly on the persistence of these paths." So his intuition was that memories are formed by creation of paths and strengthened by making these paths stronger and more persistent. And that's an idea that's been incredibly influential and really underpins a lot of our thinking today. So the experimental study of memory began with Pavlov, or at least around the time of Pavlov. And Pavlov is famous for his development of this notion of associative learning, what he called conditioning, and we still call it conditioning today. So he was actually a digestive physiologist. He was trying to understand salivation in dogs, and he was using food to elicit salivation. And he noticed that over the course of an experiment, as the dog became used to the experiment, it started to salivate not when the food was presented, but when the sounds of the food being prepared were presented. So the salivation started, at the beginning of the dog's experience, at the moment of food, and came to precede the food. And Pavlov recognized that this was something that had changed in the dog's brain, so some change in its memory had taken place. And he realized that this was a really useful way of conceptualizing memory and of making it tractable to study. So he formalized the study of this, and he developed methods for presenting stimuli and recording salivation and looking at how the association between the new stimulus and the salivation developed. And this type of very simple connection formation we call Pavlovian conditioning or classical conditioning, because it was really the first kind of conditioning to be studied. Now, the insight of Pavlov that there were connections forming between the preparation of the sounds of the food and the arrival of the food, he recognized that these had to be in the brain somewhere, and everybody else did too. And so the question became, where in the brain are these connections being stored, these memory traces, or what we call the engram of the experience. So one way of investigating this is to condition an animal, so allow it to form associations between a stimulus and another stimulus, and then to make some type of interruption in the brain in some of its connections. So one of the people who did a very extensive set of experiments along these lines is Carl Lashley. And what you can see in the slide is it's a schematic of a dog brain, and he did many experiments making knife cuts. So those red lines are knife cuts, on different animals and different experiments, training the animal, developing the conditioned response, the new response, and then making a knife cut to try and abolish that response. And he never really succeeded in obliterating the conditioned response with a single knife cut. And when he summarised all of experiments towards the end of his career, he wrote, "I sometimes feel in reviewing the evidence on the localisation of the memory trace, the engram, that the necessary conclusion is that learning just is not possible." Now, of course, we know learning is possible, so I mean, he wasn't being serious, but his point was that it doesn't seem to be an easy thing to pin down. And he actually concluded that memories are stored in a distributed fashion throughout the brain. He developed this into a kind of a principle or law, the law of mass action, which postulates that certain types of learning are mediated by the cerebral cortex as a whole, so not within a single part of it, but everywhere, contrary to the view that every psychological function is localized at a specific place on the cortex. So there's this kind of conflict between the localizationist view of memory and the distributed view of memory. And this is a conflict that's been going on for very many decades, because some experiments seem to show one thing and some seem to show another. Now, in support of the distributed idea, the very first computational neuroscientists were starting to think about how memory could, in theory, be stored in networks of neurons. And we'll be looking in this in more detail later in the course. But I just wanted to introduce you to Hebb, who was a psychologist and theorist who did a lot of thinking about these ideas. And he kind of developed this way of conceptualizing memory as what he called a cell assembly. So a cell assembly is a group of synchronously active neurons in the brain. So what this slide is showing is a schematic of how you might think of that. So let's say you want to represent different concepts in the brain. You want to represent a pineapple, you want to represent an apple, you want to represent Isaac Newton. They're all completely different things, and each one of these concepts will be represented by a group of neurons that are simultaneously active. And Hebb defined these as a cell assembly and sort of suggested that these could be distributed, right across the brain, and they could be overlapping. So, for example, the apple and the pineapple assemblies might overlap to some extent because they have some features in common. They're both fruit. They both have apple in the name. There might be links between the apple and Newton because we have an associative connection between apple and Newton and the apple that fell on his head and so on. So you can have these cell assemblies that are distributed but overlapping, and this could be a way of packing a whole bunch of memories into a smaller set of neurons in this distributed fashion. So the distributed nature of memory was a powerful idea, and together with Lashley's law of mass action, it began to seem like memory really was a distributed function spread throughout the brain. But then in the 1960s, some new evidence came along that seemed to support the localizationist view of memory, and we're going to look at that in the next video.